Imagine for a second that you survive a massive heart attack. Like, it's a terrifying experience, obviously, but you make it through.
Yeah, you survive, but the heart itself doesn't come out unscathed.
Right, exactly. During the event, a part of your heart was starved of oxygen, and that specific tissue died. And right now, sitting in your chest, that dead tissue is turning into this stiff, permanent fibrotic scar.
And it doesn't beat, it doesn't pump, it basically just sits there.
Yeah, an inflexible patch on a moving organ, which, slowly contributes to heart failure over time. But what if doctors could inject a microscopic code directly into that scar? Like a biological spell, basically, turning that dead, stiff scar tissue back into healthy, synchronized beating heart muscle.
I mean, it sounds like pure science fiction, right? We're so used to thinking of scars as the absolute end of the road, like a permanent monument to an injury.
Yeah.
But biology is, well, it's far more malleable than we give it credit for, and that is exactly the frontier we're standing on today.
Which brings us to the show. Welcome to this deep dive. Today we're exploring a just breathtaking 2026 paper published in the journal Cell Stem Cell by Kai and colleagues.
It really is an absolute masterclass in pushing the boundaries of regenerative medicine.
Totally. So our mission for this deep dive is to unpack exactly how these scientists used a revolutionary genetic screening technology to hunt down the hidden molecular villains, the things stopping the heart from regenerating inside a living body.
And incredibly, we're going to explore how they found that manipulating a surprisingly simple mechanism, one involving a basic nutrient you literally have in your kitchen right now, can force a broken heart to heal itself.
I can't wait to get into this. But to really grasp the magnitude of this breakthrough, I think we have to look at the baseline biology of the mammalian heart, right? Which is notoriously stubborn.
Oh, incredibly stubborn. I mean, if you look at certain amphibians, like, say, a salamander, if it loses a limb, it just grows a new one.
It just knows what to do.
Right. The cells know how to regenerate.
Yeah.
But if you damage a mammalian heart, it has almost zero regenerative capacity.
Yeah, our bodies just hit the panic button. Post-injury, these cells called cardiac fibroblasts, they basically rush into the damaged area.
They're essentially the body's emergency responders.
Right, and their only goal is to rapidly lay down scar tissue to prevent the heart wall from, well, physically tearing open.
Which is a phenomenal lifesaver in the short term. It keeps you alive right after the attack. But it's a total disaster in the long term because that scar tissue, it can't contract.
It's just dead weight.
Exactly. So for years, the dream of the cardiovascular field has been something called in situ direct cardiac reprogramming.
Okay, break that down for us.
The core idea is to take specific transcription factors. Think of them as master control switches for a cell's DNA. We're talking about the MGT cocktail, so things like MEF2C, GATA4, TB/5, plus myoford and cell4.
That's a lot of alphabet soup, but basically it's a reprogramming code.
Right. You use this cocktail to force those stubborn, scar-forming fibroblasts to change their identity. We want to overwrite their programming and turn them into what we call induced cardiomyocytes, or ICMs.
So in plain English, turning scar cells directly into beating muscle cells, right there in the heart.
You nailed it.
And scientists actually have this cocktail. It acts like a new operating system for the cell.
Yeah.
But, and this is where I hit the central mystery when I was reading the sources, if you take these scar fibroblasts out of the body, put them in a nice, safe Petri dish, and give them this cocktail, it works beautifully.
In vitro, it's fantastic.
Yeah, the cells turn into muscle. But when scientists try to do the exact same thing inside a living post-heart attack mouse, inside the in vivo environment, the efficiency completely plummets. It barely works.
It's a huge bottleneck.
Why is the living body so hostile to a cure?
Well, it comes down to the environment. What's fascinating here is that a heart right after a myocardial infarction isn't just a static, quiet wound. It's a biological war zone. You have a raging immune response, massive waves of inflammation, the physical extracellular matrix is rapidly changing, and those myofibroblasts are hyperactive.
So I was thinking about this, and it's basically like trying to bake a delicate Michelin star souffle, which is our reprogramming process, while standing in the exact center of a Category 5 hurricane.
That is the perfect analogy. You might have the perfect recipe, but the environment is violently ripping your ingredients apart before they can even mix.
So the hurricane itself actively fights the reprogramming. Let's do a quick recap of this first part for everyone listening. Bullet 1. Heart attacks cause permanent scarring via these emergency responder cells, the cardiac fibroblasts.
And bullet 2. We have the genetic tools to reprogram those scars into muscle, but the living body vehemently rejects the process.
Right, which leaves you with a question to ponder. Have you ever wondered why a simple cut on your skin heals flawlessly, but your heart seems hardwired to self-destruct with scar tissue?
It's a bizarre evolutionary trade-off. We prioritize a quick, sloppy patch over functional regeneration.
So, to fix this souffle and hurricane bottleneck, the researchers needed to figure out what was ruining the bake. They had a list of about 140 different genes that might be acting as barriers.
Yeah, 140 suspects.
But how do you test 140 genes at once in a living, beating, inflamed heart? My immediate thought was, well, we have CRISPR. Why not just inject viruses carrying CRISPR directly into the heart, knock out all 140 genes in different cells, and see what works?
It's a very logical thought, and it's the traditional approach for a lot of gene therapy. But in this specific post-MI environment, direct viral injection is incredibly dangerous.
Why is that?
First off, it's messy. Viruses don't just politely enter the exact cells you want them to. They infect the wrong cells, creating off-target effects that completely muddy your data.
So you wouldn't know if a cell failed to transform because of the gene or just because the virus missed it entirely.
Precisely. But more importantly, there's a fundamental issue with how the CRISPR system operates. It works by creating actual physical double-strand breaks in the DNA.
Oh, right. It literally cuts the DNA.
Exactly.
Yeah.
And in an already dying, stressed-out heart, the cellular toxicity caused by intentionally breaking DNA strands. It pushes the cells over the edge. It literally makes the heart attack worse.
Wow. Okay, so you're trying to cure the heart by essentially nuking it with DNA damage. That's obviously not going to work. So how did Kai and the team bypass this? Because their solution, this in vivo Perturb-seq using cell transplantation, is just brilliant.
It's a total masterstroke. They entirely inverted the problem. Instead of injecting chaos directly into the heart, they pulled the target cells out of the chaos.
They extracted cardiac fibroblasts from a mouse that had already had a heart attack.
Right. They took the soldiers out of the war zone, put them in a dish, but they didn't just mix them all together in a big soup. They used an arrayed format.
Meaning they separated them into a massive grid of individual wells.
Exactly. And in each separate well, the cells received the reprogramming cocktail, that MGT operating system, plus just one specific genetic dimmer switch.
And these dimmer switches aren't CRISPR, right?
No, they used short hairpin RNA, or shRNA. It doesn't break the DNA. It just intercepts the instructions for a specific gene and silences it or knocks it down.
So by keeping them in separate wells, they guaranteed every cell got the muscle instructions, plus exactly one of the 140 suspect genes turned off.
They created a perfectly controlled starting line. Then they pooled all these beautifully prepared cells together and transplanted them back into the damaged border zone of a living mouse's heart.
But wait, if they put them back, wouldn't the reprogramming start immediately before the cells even settled into the tissue?
That's the genius part. They engineered a delay. They made the reprogramming cocktail completely inactive until it was exposed to a specific trigger.
Oh, the doxycycline diet.
Yes, they waited for the cells to fully engraft into the hostile heart tissue. Only then did they feed the mice doxycycline, which acted as a chemical key to turn on the reprogramming.
So they let the cells face the hurricane, flip the switch, wait 7 to 14 days, and then pull them out. And they could find them because they tagged them to glow fluorescent green EGFP.
Exactly. Laser sort the green cells, run single-cell RNA sequencing, and see which hRNA survived the hurricane best.
Let's quickly summarize that brilliant workaround. Bullet 1. Traditional in vivo CRISPR screening is too toxic for a damaged heart.
And bullet 2. They use a take-out, modify, put back cell transplantation strategy activated by doxycycline to test 140 barriers safely.
Right. And it makes you think by removing the cells from the chaos just long enough to program them, they created a standardized starting line. How often we fail to solve a complex problem in our own lives just because we haven't isolated the variables?
It's a great lesson in problem solving. So now they have this mountain of sequencing data. Every cell has a barcode telling them exactly which gene was turned off.
Right, and to make sense of this, they used a technique called single-cell trajectory mapping.
Which sounds incredibly intimidating.
It does. Well, let's visualize it. Imagine a sprawling highway system. Every cell starts its journey at Fibroblast City.
I love this.
Their destination, driven by the cocktail, is cardiomyocyte town. But because of the hurricane of inflammation, the road signs are destroyed. The sequencing GPS showed most cells got totally lost.
Yeah, taking the wrong off-ramps into dead-end wastelands, like becoming entrenched matrix fibroblasts or getting bogged down in the interferon response suburbs.
Right, just fighting inflammation forever.
Right.
But because of the barcodes, they could filter the data to look only at the cells that actually arrived at cardiomyocyte town. And they basically asked, what was your unique advantage?
And out of 140 targets, one stood out drastically. Knocking down a gene called Calr resulted in the highest enrichment of cells actually making it to heart muscle.
Calr was the ultimate villain. What does it actually do?
It codes for a protein called calreticulin. Now, calreticulin is an endoplasmic reticulum, or ER resident chaperone. The ER is the cell's internal manufacturing facility.
Okay.
Under normal conditions, calreticulin acts like a highly absorbent molecular sponge. Its main job is to hoard calcium inside the ER, keeping it locked away.
Wait, a calcium sponge? The biggest barrier to healing a heart attack is just a protein hoarding calcium.
It seems strangely specific, doesn't it? But look at the clinical connection. The paper highlights that Calr is massively upregulated in infarcted hearts, and not just in mice.
Oh, really?
Yeah, they cross-reference this with human spatial multi-omic maps, the Kupi et al. Dataset. The acute stress of a human heart attack actually causes the heart to panic and overproduce this calcium sponge.
Wow. So to recap segment 3, bullet 1, single-cell trajectory mapping showed the exact fate of every perturbed cell.
And bullet 2, calreticulin. That Cal gene was identified as the absolute top barrier to cardiac reprogramming.
Which raises an audience question. If a protein's normal job is just to manage calcium, why would a heart attack cause it to go into overdrive and block healing?
It's a desperate defense mechanism that becomes the very roadblock halting regeneration.
Okay, let's unpack this next part because I really want to understand the how. What exactly happens inside the cell when this sponge is removed? Give us the step-by-step molecular chain reaction.
All right, the mechanism is beautifully elegant. Step 1, you use the shRNA to knock down Calr. The sponge vanishes from the ER.
So all the calcium it was hoarding, where does it go?
Step 2, calcium is no longer trapped, so it floods out of the ER and spills directly into the cell's cytosol, the main fluid.
Just a massive spike of free-floating calcium.
Yeah, exactly. Step 3, this huge calcium spike binds to a specific sensor protein floating around called calmodulin, or CAM.
Okay, calcium hits calmodulin, then what?
Step 4, calmodulin activates a specific kinase enzyme called CAM-KI delta. And step 5, the grand finale. CAM-KI delta physically phosphorylates MEF2C.
Wait, MEF2C, that's one of the master transcription factors from our original reprogramming cocktail.
Yes, we already gave the cell MEF2C, but this calcium cascade supercharges its transcriptional activity.
Oh, wow.
It's so powerful that the researchers proved knocking down Calr can completely substitute for adding MEF2C to the cocktail. The calcium spike wakes up the cell's natural MEF2C.
That is just a staggering domino effect. But how do we know this is definitely the mechanism?
The scientific validation is rigorous. They ran 2 tests. First, they used a chemical called FPL64176.
What's that?
It's a calcium channel agonist. It artificially floods the cell with calcium, and it perfectly mimicked the Calr knockdown regenerative effect.
It proved calcium was a trigger.
Right. Then they used BAPTA-AM, which is a calcium chelator, basically a microscopic vacuum that instantly sucks up all free calcium.
And let me guess, it completely blocked the regeneration.
Completely ruined it. The evidence is airtight.
That's incredible.
And there's a deeper developmental link here. If you look at how an embryonic heart forms, it relies on calcium oscillations to start differentiating and beating.
Wait, so by removing the sponge and flooding the scar cell with calcium?
We're literally tricking the adult scar cell into thinking it's an embryo again.
That gives me actual chills. Okay, let's summarize this calcium cascade. Bullet 1. Suppressing Calr releases a flood of calcium into the cell's cytosol.
Bullet 2. That calcium activates the calmodulin and CAM-MKI delta cascade, which supercharges the MEF2C protein to drive heart muscle creation.
So we usually think of complex genes driving biology, but here, a simple ion calcium holds the keys to the kingdom. What other fundamental ions might be secretly dictating your cellular fate right now?
It's a profound thought, but, a beautiful mechanism is only half the battle.
Right. Does tricking the cell into thinking it's an embryo actually translate into tangible healing for a damaged heart? Let's get to the results.
The Petri dish results were amazing. Under the microscope, the newly reprogrammed cells didn't just look like muscle, they showed synchronized calcium oscillations.
Meaning they were literally beating together in a coordinated rhythm in the dish. But what about in vivo? in the living mice.
14 days post-MI, treating the heart with the reprogramming factors plus the Calr knockdown, they called it TDMGT, plus Calr, yielded staggering results. Up to 16.5% of the targeted cells converted to heart muscle.
And they verified that by checking if they were CT9 positive, right?
Exactly. And 16.5% is a massive yield in living tissue.
And the physiological data backs it up. They did echocardiograms on the mice. The ejection fraction, how much blood the heart pumps, hit 33% in the treated mice, compared to just 24% in the controls.
The pumping capacity was fundamentally restored.
And they used Masson's trichrome staining to look at the physical scar. The fibrosis in the ventricular wall dropped from 51% down to 43%. The scar was physically shrinking.
But here's why this matters for the listener. This wasn't just a mouse study.
The ultimate test. Human translation.
Right. They isolated primary cardiac fibroblasts from a 48-year-old human heart transplant recipient.
Actual human heart attack scar tissue.
Exactly. And knocking down CALR in these human patient cells increased the generation of new heart muscle cells by 12-fold.
A 12-fold increase. That is unbelievable. So recap 5. Bullet 1, Calr knockdown created functionally beating synchronized heart cells.
Bullet 2, in living animals, it restored pumping capacity and visibly shrank the scar.
And bullet 3, it successfully reprogrammed actual human heart attack scar tissue.
It proves the human heart inherently wants to beat again. It just needs the calcium lock removed to remember how.
This journey has been incredible. We started with the grim reality of a permanent scar. We saw how a cellular transplant screening method bypassed the toxic environment.
We found the hidden villain, calreticulin.
Right, and revealed how a temporary flood of calcium reboots the embryonic code, turning a scar into beating muscle. Why does this matter to you listening? Heart failure is one of the greatest global health burdens. We're looking at a paradigm shift where your own pathological scar tissue could become the very medicine that regenerates your heart.
In this deep dive, we learned the adult heart still has the genetic memory of how to grow and beat like an embryo. It was just locked behind a calcium sponge.
That's such a powerful concept.
So as you go about your day, it raises an incredible question for you to ponder. What other dormant, miraculous developmental superpowers are hiding inside the useless or damaged tissues of your own body, just waiting for the right molecular trigger to wake them up?